In this video lesson, we're going to talk about the transport of materials, specifically the transport of materials across a cell membrane. Cell membranes are said to be selectively permeable. So when you're being selective, you choose what you want. So cell membranes choose what they want to be allowed in or out of the cell. So permeable means that it's open to letting things cross across the barrier. So nutrients, water, and waste need to be able to get in and out of the cell, but we don't want all of our nutrients exiting. We don't want all our water entering and then causing the cell to burst. So cells need to be selectively permeable, and there are some ways that it gets around that. Diffusion is when molecules move from an area of high concentration to an area of low concentration. And what happens is they keep moving across the membrane from the area of high to the area of low until they are balanced across both sides. So what's happening is the molecules that are in that area where the concentration is high are bumping into each other because they're more crowded together. And when they bump into each other, some of them will get pushed into the area where the concentration is low. And eventually they'll be evenly spaced across the cell membrane. This is a visual of what that looks like. So here are all these molecules in an area of high concentration, and they're jostling each other and they're getting bumped. And eventually some of these are getting pushed to the area where it's low. And over time, you'll end up having a balance between the two sides. So some examples of diffusion in the body. So when we breathe in a mixture of air that has some oxygen and carbon dioxide and other, mostly actually nitrogen, but there'll be a higher amount of oxygen in the air sacs of our lung, and it'll get absorbed into the bloodstream because it needs oxygen. It's used it up through the cells. And then the carbon dioxide then on the same way, they'll diffuse out of the blood that's near the lungs and into the air sacs, and then we'll breathe out the extra carbon dioxide. And the same thing happens at our cells. As the cell uses up oxygen, it will pump out carbon dioxide as a waste product, and then it'll get carried to the lungs to get rid of. And then when we digest food, the nutrients will diffuse into the bloodstream from the small intestine so it can be carried and dropped off to different cells. Some everyday examples of diffusion would be when you're making a cup of tea. So you put a tea bag in a hot cup of water, the tea particles will diffuse out and they'll spread out through the whole cup, and eventually you'll have a nice cup of tea or coffee, whatever you're making. It happens better in hot water because the particles are moving faster, and that's why we use hot water to make tea and coffee. A real life example actually happened in class once when somebody released a fart, and the fart particles traveled throughout the classroom. So eventually the entire classroom smelled like the fart. And nobody noticed at first, only the people that were in the area of high concentration where the source was. And eventually those particles spread through the entire classroom. Osmosis is when water moves instead. So sometimes not all particles or substances can go across the cell membrane. So osmosis will take place, and that's the diffusion of water molecules across the cell membrane to balance out the concentration of a substance across the cell membrane. So if, for instance, maybe sugar can't get out of the cell, but there's too much in the cell, water will go in to dilute the sugar so the concentration is the same on both sides of the cell. There are three terms that we need to know when we're talking about osmosis: hypotonic environments, isotonic environments, and hypertonic environments. So the first one is the hypotonic environment. So what that means is the concentration of the solute in the cell is higher than the surroundings. So there's more, we're talking about sugar or salt, there's more sugar in the cell than in the surroundings. So water will move into the cell to balance out the concentration of the solute. this is what it looks like. So there's a diagram that I took from our textbook. So the hypotonic solution, water is going in because there's not as much, but it's not really showing the particles. So I did the same thing over here. So here's my cell. It's got a whole bunch of particles inside the cell and only a few of these little particles in the surrounding area. So what happens is water starts to go in like so, and then the cell will swell up. And if too much water goes in, the cell will actually explode. So this is a red blood cell. Normally, we say it has a biconcave shape. It basically looks like a donut, but the hole doesn't get punched in all the way. So it's not completely hollow in the middle. If too much water gets in, it blows up like a beach ball and actually could explode if too much water gets in. This is a plant cell, so because it has a cell wall, it can't grow and shrink in the same way, but same thing, not as many particles here, so the water goes in, and then the cell membrane will push on the cell wall and cause it to be turgid, so it'll be rigid and look a bit swollen. An isotonic environment is what cells want. They want the same concentration of solute inside and outside of the cell. When a cell is in isotonic conditions, there's no, they say, net movement of water. So there might be one water molecule that will leave the cell, but then another one will go back and enter the cell to balance out the concentration. So everything's equal on both sides of the cell. So it just looks like this. So if one water molecule leaves, one comes back to replace it. It doesn't change. The cells look like they should. They're in their regular format because there's no shrinking or swelling of the cell because everything is balanced. Our last scenario is a hypertonic environment. So when a cell is in a hypertonic environment, the concentration of the solute in the cell is less than the surrounding. So that would be like if you put a cell into like salt water, that would be not so good for the cell if it's a high concentration of salt. So water will have to move out of the cell to dilute the solution that's outside of the cell. So again, here's our diagrams. This is my cell. There's not much solute in the cell. There's a whole bunch of particles outside the cell, so water is leaving the cell. So it looks like a balloon that's deflated. It's all shriveled up. And then same thing for the plant cell. So water is leaving, and then the inside of the cell literally like shrivels up, but then, and the cell wall is still there, but it's kind of collapsing on itself too. I have a real life sad story about a hypertonic solution or environment. When I was 5, I had a goldfish that I got, and I thought my goldfish bowl looked a little sad, so I decided to take some seashells that had been collected when there was a trip to Florida, but seashells are from the ocean, so they were covered in salt. So I put them in the goldfish bowl, and then the salt dissolved and was in the goldfish bowl, and then the fish was now in a salty environment, and that wasn't good for my fish. So the water left my fish, the fish shriveled up and died. And I didn't know until I learned about osmosis that I myself had actually accidentally caused the death of my fish. I thought it just died of natural causes, but I was wrong.